Hello, good afternoon, everyone. Good morning. This is webinar 2 of the fifth NOPAP training course on remote sensing data analysis. We have many people coming in this room. My name is Genki Terauchi. I work for NOPAP CILAC since 2004. And this is our very first webinar dedicated to monitoring and assessment of water quality by ocean color remote sensing. To start with, I would like to introduce the website we have developed for this webinar. Please find the link to the web page that I share on the screen. You see the schedule for the webinar too. And now it's 2 p.m. in Japan Standard Time. And so we will be soon beginning the day one. So all the information you need during the webinar is aggregated on this website. So if you get lost, please always go visit this website. All the information resources you need during the course is available here on this website. So, and if you have any problem accessing this website, please let us know. So I will proceed according with the program. First, I would like to introduce the director of NOPAP CILAC, Mr. Makoto Hayashi, to give the opening speech.
Good afternoon, everyone. I am Makoto Hayashi, Director of NOPAP CILAC. Thank you very much for joining the 5th NOPAP training course on remote sensing data analysis, webinar 2, monitoring and assessment of water quality by ocean color remote sensing. Firstly, I'd like to express my thanks to all organizing committee members, all lecturers of this training course, and CILAC staffs. Now, I'd like to briefly explain our organization, NOPAP CILAC. The full name is the Special Monitoring and Coastal Environmental Assessment Regional Activity Center of the Northwest Pacific Action Plan, NOPAP in short. Actually, NOPAP is a part of the Regional Sea Program of United Nations Environment Program, UNEP. There are 4 regional activity centers in the 4 member states: China, Japan, Korea, and Russia. And CILAC is a regional activity center in Japan. The center is located in Toyama Prefecture on the middle coast of Japan. Mandate of CILAC is assessing the state of the coastal and marine environment. And we are also expected to develop new assessment tools and methodologies using the state-of-the-art technologies such as remote sensing. Capacity building is another important role for CILAC to facilitate application of new skills in the NOPAP region first and hopefully worldwide later. Therefore, we have conducted the training course on remote sensing data analysis 4 times from 2007 to 2013 in Japan, Korea, Russia, and China. And 91 people from the NOPAP region and neighboring Southeast Asian countries joined the courses. We are proud of saying that the high percentage of those trainees are still engaged in related fields in their work. Now it is a new era of remote sensing since people started using Google Earth Engine. cloud-based platform for processing a large amount of satellite images and data sets. CLAC was very quick to take advantage of it in our assessment projects and constructed some assessment tools. Eutrophication is one of the major problems in the coastal areas worldwide. CLAC launched activities on eutrophication in 2008 and developed the common procedure for assessment of the eutrophication status for the NAUPAP region in 2009. The methodologies consist of two steps: screening procedure with a minimum number of indicators and comprehensive procedure for more thorough assessment. In 2019, CLAC improved the screening procedure and developed the NAUPAP eutrophication assessment tool, so-called NEAT. Then we refined the tool to be applicable worldwide as Global Eutrophication Watch. NEAT and Global Eutrophication Watch will be introduced in the lectures and hands-on sessions in this webinar. By the end of this training course, I hope all of you will be familiar with these tools and be ready to use them in your current and future research activities, and share the knowledge and skills with your colleagues as well. At the end of my welcome speech, thank you again for your dedication to the conservation of the marine environment, and have fun at the training course. Thank you very much.
Thank you very much, Director Hayashi. So today, day one, we have one lecture and one hands-on session. Please be noted that the link to the webinar and the lecture and hands-on sessions are different. And as I said, the information is made available on the GitHub account, GitHub website, available from the chat. Okay. So I would like to proceed according with the program. We have, as a first speaker of the day, we have, I see someone's raising hands. Hello, Henri Cabe, if you want to talk something, just please interrupt me. I see you raise a hand. Okay, it was your mistake. All right, our first speaker of the day one is Dr. Joji Ishizaka. He is a professor at the Institute for Space and Earth Environment Research, Nagoya University. Dr. Ishizaka has been supporting the NAUPAP program since the very beginning of this program. He was a member of NAUPAP Working Group 4 that was trying to develop a regional monitoring program using ocean color data. And since 2008, he has been supporting NAUPAP activities of NAUPAP CLAC as our representative of Japan, as a CLAC one of the CLAC focal point member. And so his dedication to this NAUPAP program is exceeding more than 2 decades. So now I would like to welcome Dr. Ishizaka to give the very first lecture of the day one. The room is yours.
Okay, good afternoon. Thank you, Genki, for the introduction. Okay, so my name is Joji Sutaka. I'm from Nagoya University, Japan. And so today I'm going to have the one lecture about the biological oceanography and the ocean color remote sensing. So I will share the screen. Uh, can you see? Okay, I believe you can see the slide. And so I have only one lecture, which is 40 minutes, I believe. So it is not very long. And but the contents about the satellites of biological oceanography and ocean color remote sensing, this is a kind of very, very broad and wide thing. And if I can spend maybe maybe a couple of weeks to talk about, but today I'm mostly focusing on some points and which is here about the unification, global distribution, and of course satellite detection, the chlorophyll and seasonal variation, primary production, change in efficiency, and phytoplankton group identification. And this is already too much for one one lecture, I know. So today's lecture is kind of very short introduction. So those you know this field already, maybe this is just a reminding of your knowledge. And those who do not know this kind of things, you probably need to study more, maybe using the textbook of biological oceanography or maybe satellite oceanography. Yeah, so that's you need to do. Okay. Okay. First of all, yeah, eutrophication maybe covers some other lecture or hands-on, of course, but this is one of the most important topic for NAUPAP, and yeah, this kind of thing is well known long time. Okay, maybe some of you notice looking at the coastal area or maybe lake, the water color looks like those. Okay, sometimes the red, green, or sometimes red, and it is not really blue ocean. Yeah, so which things which might be indicating there are some pollution, and it is, and sometimes those kind of phytoplankton bloom, which is meaning of the large amount of the phytoplankton in the grown in the water. And then there might be some cases there's a fish kill. Yeah, some things happen. And this kind of problematic algal bloom is called harmful algal bloom. And this will cover tomorrow's lecture by Wonkook Kim. So I don't say so much. But actually, this kind of thing, one of the reasons, of course, there are probably many reasons, but one of the reasons may be eutrophication. Okay, so this is a very schematic figure of the coastal ecosystem. So in coastal area, we probably have a nutrient organic matter inputs from maybe land to the water. And then this kind of nutrient can be used by the phytoplankton. And phytoplankton is kind of primary producer. So this is the first organic matter and eaten by zooplankton. And the zooplankton should be a good prey for the fish. And hopefully the fresh fish increase, then we can catch the fish from the coastal area and then we can eat, okay? So if there's this kind of the material cycle or energy cycle in the coastal area, it will be very nice, okay? So especially if we can increase the nutrient and if we can increase the fish amount, that will be very nice. But unfortunately, often it is not the case that if we increase the nutrient or organic matter input by the anthropogenic human activity, then what will happen is like this. So if we put the pollution of the organic matter or nutrient, then the phytoplankton usually the abundance of the phytoplankton increase so much, and often produce say like a red tide. Yeah. So this is the phytoplankton bloom condition. And then also the phytoplankton, this is kind of organic matter. So the organic matter is actually usually decomposed in deeper water. And then for the decomposition of the organic matter, the oxygen will be used. Okay, so this kind of things often makes a low oxygen condition. And sometimes we call dead zone, yeah, because for the low oxygen condition, all the animals like zooplankton or fish cannot live, okay? And of course, if we don't have fish, then there is no fish catch anymore, right? And of course, and also if there is some, the aquaculture, maybe fish kill is a very serious problem. And this kind of low oxygen condition, sometimes water is coming up and become blue, yeah, because of the hydroxide sulfur makes this kind of blue color. So this is another water color change, yeah. And it is also known that if phytoplankton increase so much, maybe seagrass or seaweed, which is very important for local production, is also decreased. And this seagrass seaweed was covered in the webinar, first webinar, I believe. Okay, so this kind of thing is worldwide happen, and it is in the SDGs, we have a 14 life under the water, and the 14.1.1a, it is specifically talks about the index of coastal eutrophication, which is a very important aspect. Yeah. But of course, this is not, in some area, actually, there are some difference. And in Japan, this kind of thing is known for a long time, yeah. So we actually makes a very effort to decrease the input of organic matter or nutrient to the coastal water from the river, yeah. And now actually some people think it is too clean, okay? So too clean means there's a low phytoplankton. And if the low phytoplankton, there's a low zooplankton or fish, yeah. So this may be another problem, so which is called oligotrophication, which is the inverse of the eutrophication. Yeah, so there are some such problems is known in some area now. Yeah. So I'm going to talk about the very large view of the primary production of phytoplankton distribution. So this is the ocean color distribution, ocean color view of the phytoplankton. So you can see there are some areas blue, which means a very, very low phytoplankton. And actually, and some areas green or red, which is a higher phytoplankton, which is actually known as a good fishing ground usually. Yeah, so it is kind of controversial or contradicting that eutrophication is a big problem, but we need some nutrient input to the ocean, so that we should have a high production ocean. Yeah, so in most of the area, actually, this kind of distribution is very much corresponding to the wind-driven surface currents condition like this. Yeah, so this is called Jaya, which is a very strong current in the oceanic area, and it is known that the very nutrient is low and the plankton is low. Okay, and there is less fish in here. Yeah. So we know 70% of the ocean, I mean 70% of the Earth is covered by the ocean, but the very large area of the ocean is actually low production. Yeah. So this is another, maybe in future, maybe we need to think about how to use those kind of area. Yeah, but in any case, and later I will talk about this maybe also becoming even low production. And in here, yeah, looking at the vertical section here, no, before that, yeah, and this kind of distribution is actually known quite a long time ago, 1955. Yeah, there's a physical oceanographer. He is actually expect this kind of phytoplankton distribution, higher production area, which is very much similar to what satellites can see now. Yeah, but of course, 1955 we didn't have a satellite. But he could expect this kind of distribution from the point of physical oceanography point of view. Yeah, and he was expected from the current surface current condition that those area is high production area is high nutrient condition, and the low production area is low nutrient condition. Yeah. Okay, so it is very much depending on the physics is very important for the large scale of the ocean. Yeah, so this is the temperature and the phytoplankton distribution, and this is Japan actually. So this is a satellite put together with a section of the ocean from surface 300 meter. And you can see the warm water is actually floating in this southern area, actually tropical low latitude area. Yeah. So, and this area is very, very low nutrient condition. And you can see phytoplankton amount is very low in here. This pink color is a low phytoplankton. And of course, there is some kind of deep phytoplankton maximum here, which is called subsurface chlorophyll maximum, which is kind of between the low nutrient, high light condition surface water to the deep, very low light condition with high nutrient condition in here. Okay, so this is kind of interesting to see. Nutrient is actually high in the deeper water, which is the next slide here. So this is the same section of what I showed before. And here is the phytoplankton. And the phytoplankton is surface for the higher latitude here. And, but it's become deeper in here. And above this high chlorophyll maximum, actually the nutrient is very, very low. Yeah, it's like less than 1 micromole per kilogram for nitrate. Yeah, which is very low. Yeah, and the deeper area, actually there is a nutrient. So if the most of the ocean, this nutrient is deeper nutrient is a source of the production. Yeah, like here in the subsurface. Yeah. But of course, in the yeah, and it is also interesting to see this. This is total carbonate, CO2, total CO2. Yeah, so this is not only molecular CO2, but including ion of carbonate material, which is the distribution is very, very similar between the nitrate, because the biological production is influenced similar way. But actually there is still the magnitude is much higher for total carbonate, because total carbonate is source is atmosphere, and there is a very high abundance in the water. Yeah. And so this is also related to carbon cycle. I will talk about this later. But the important thing is why this kind of two-layer structure happened. nutrient can be in the upper area here, but actually, if there is a nutrient in upper layer, phytoplankton should be able to use nutrient and grow. But then eventually you will sink to deeper water. Yeah, so that makes this very low surface water. But then why not in the northern area? Yeah, this area is actually well mixed from the deep water to the surface because very low temperature area. Yeah, so mixing is a very important source of nutrient to the surface area. Yeah. So how do you know that the material is sinking down? Okay, so there's some equipment called sediment trap. And this kind of equipment is putting in a deeper area. And then this is actually 1 month for one bottle. And so within 1 month, a lot of particles is sinking down from the surface of the ocean to this equipment. Yeah, this equipment probably putting on about 1,000 meter deep of the ocean. Yeah, so then after 1 month, we can see a lot of materials. So this is coming from the sea surface, which after the production. Yeah. Okay, so this kind of thing is called biological pump. Yeah, so this is very important for both nutrient dynamics or carbon dynamics. Yeah. Okay, then what is the controlling factor of phytoplankton growth? Yeah, it is well known the nutrient is important, and which is material source. Yeah, and the light is of course because of primary production, phytoplankton is a primary producer. Light is also important, and this is energy source, right? And temperature also may be important, but this is only the rate controlling factor. Yeah, so usually the nutrient light is the most important thing. Yeah, temperature may be important for the phytoplankton species level. Yeah, very important. Okay, and of course those things also controlled by something. And as I said, physical factors is very, very important, including the wind or current. Was stratification. Stratification is how much water is separated from the surface to deeper area. Yeah. And of course, even in the larger, in the very far from the coastal area, actually, there is influence of the land through, say, yeah, the river, if close to the coast, yeah, it should be the river. But there is another source, it's called aerosol, which is material from atmosphere. Yeah. And then the influence of climate change, anthropogenic factor, those are the very important factor for controlling those things. Yeah, so so we should understand what this kind of things deeply. Okay, I don't have much time now, I understand. Okay, for the ocean color, so we will see the ocean color using energy of the sun, and the sun energy coming down to the ocean and makes a color of the water, and then the sunlight can detect this. But in the water, there's some material scattering, light scattering or light absorption properties inside. Yeah, like a suspended matter or phytoplankton or dissolved organic matter is the source of those light change or color change. Yeah, so if we know the color of the water, we should be able to understand the concentration of those materials. Yeah, but we should be really careful about using a sunlight because there are some large effect of the atmosphere. Yeah, even the cloud or molecular or aerosol. Yeah, so this is atmosphere is very, very important factor, and I will speak this. Yeah, about if the surface from the the atmospheric information is very large, maybe 90%, more than 90% of the signal of each wavelength. Yeah, so the water signal for the water is only this one. So we should know the water color is this part, but we need to take out those atmospheric the information. So this is not so easy things, and we should be a little bit careful about this. And especially for coastal area, we have many problems for both atmosphere or in water part. And in water, I don't, yeah, it explains so much, but besides the water, phytoplankton is of course a very important absorption material. But there is also other material like detritus or the color dissolved organic matter, yeah, which is another factor of the. So the ratio between those things is also influenced to the ocean color. So this is a simple model that if phytoplankton increase, say this is spectra, of course, the blue color decrease, so it's become more green to red, yeah. But similar thing happened actually suspended solid, yeah. This is a more scattering material, so it become more and more bright. But the slope become very similar to this one. So this is a factor, one factor of the make error. And there's also color dissolved organic matter. This is another factor which is also absorb the blue part, right, similar to the phytoplankton. So those factors are you should take into account sometimes. Yeah, hopefully someone will make a very good algorithm to take care of those atmosphere and water material other than phytoplankton. Okay. Okay, so this is a seasonal variation, and you can see up and down movement of the high chlorophyll area or low chlorophyll area. Yeah, depending on the season, it will change so much. Yeah, and the seasonality is like this in around Japan. So we have a high in spring and autumn, and phytoplankton is low in the winter and the summertime. And if I spend the time here, I can explain very much. But this is not only just a temperature effect, but it is actually physical structure mixing is very, very important. And there's so-called critical depth theory, but I don't explain so much. But essentially what happened is if phytoplankton is in the surface, bright light condition area, the phytoplankton can do the photosynthesis, so they can grow. But if the mixing is very strong, like a winter, and if the phytoplankton go to the deeper area, spend the time in a dark area, then they cannot do the photosynthesis so much, so they cannot grow. Okay, so that is the reason that there is a two. In a temporal region, there's two peaks. Okay, one is in spring and one is autumn. Yeah, in winter there's light is limited because water is mixed very deep area, and for summer there is stratification. So nutrient is depleted like in the middle of the ocean. Yeah, then the grazing maybe zooplankton can eat. And so it is decreased, yeah. So this kind of water column structure, stratification change makes this kind of blooming condition. So you should take into account this kind of seasonality for if you need to analyze the coastal, say, eutrophication, yeah. And okay, so this is a global carbon cycles things and the schematic, and I cannot explain so much in here. So, but here you can see the number of 50, which is from the inorganic carbon to the organic, the biology, yeah, the organisms, yeah. So this is 50. So this is actually primary production part. But how do you know this is 50? Yeah, of course, now we have sunlight. We do some modeling to express this 50. But there is a long history to develop this kind of thing. Yeah. So this is a chlorophyll by the measure of the sunlight, and PAB, this is a light condition, and temperature. We also can measure temperature. And from those, we can have some model to calculate the primary production. Yeah. As I said, I said light is important and temperature is important, but the nutrient is also important, as you know. But for this kind of global model, usually temperature is maybe including a nutrient condition. Yeah, that's some people do use. Of course, there are many other type of the modeling to calculate the primary production now. Yeah, it is. But for we need the measurement of the primary production, and still we are using some very classical method to develop maybe 1950s. Yeah, so using carbon 14 or 13, organic matter, inorganic matter, add those things and to incubate the water, and then measured how much carbon 14 or 13 become organic matter. Yeah, so this kind of thing is a very, very tedious thing. So also for the incubation, we need a very tedious work. Yeah, so okay. Then actually I pick up maybe history of the primary production, global primary production value. Yeah, it is starting from 1940s. Yeah, about 80 years ago. Yeah, so it is about 126 and then become very small, 15. And for those, most of the part is actually using the water sampling and then measurement of primary production, and then using some map to calculate the globally. Yeah, the very simple map. But now we have a satellite, so we can have a very detailed distribution of the phytoplankton and time change. Yeah, so we can get this model. But so this is 1995, they developed the first satellite model, but we are still developing the better models. Yeah, always research is doing next step, okay? Okay. For the East China Sea, I'd like to talk about it, but East China Sea, but I don't have so much time. But this is known, and this area is very known about eutrophication. But this is also a very difficult area of ocean color because this is very, very turbid area. Yeah, it looks like water is yellow. Yeah, because this is called Yellow Sea. Yeah. And so we developed the new method, collaboration with Korea or China, yeah, Japan. And so we took a lot of data samples and then develop the specific method to estimate chlorophyll A with accurate way. Yeah. And then we see some interesting seasonal structure. And this is in this area, very phytoplankton, this change is very much related to the turbidity. Yeah, this TSM is the total suspended matter, which is very high in the coastal area. But phytoplankton is actually very high in summer when the turbidity is not so much, and but also river discharges is very high in this time. Yeah. Okay, so also we are in this kind of interesting for, it is a bit old data, but for some area we can look at the time series, time change, and we can see in this area the phytoplankton is actually decreasing with river discharge decrease. But in this area, Yellow Sea, the actually phytoplankton is increasing, and we know in this area this is still increasing. So the increasing may be the influence of the nutrient input by anthropogenic. So as Dr. Maure will explain later in hands-on, that we developed this global eutrophication watch using this trend analysis. Yeah. Okay. So and but also it is important that if you look at this global view, there are some area which is actually low phytoplankton, but decreasing in a large area. So that means maybe in the very low phytoplankton area, the nutrient is decreasing now. Maybe this is related to the climate change issue also. Yeah. And so, okay. Okay, I think I need to stop here. But also we know there is a phytoplankton is different in a different region, and it is also very important because different phytoplankton makes a different food chain structure, and so we also maybe the global warming changing this condition, not only the production in the phytoplankton biomass, but also the phytoplankton group also change, and that may change the structure of the food web. Yeah. So this is an important topic, and yeah, this is the last slide that yeah, this kind of,
you can continue to the end.
End? Yeah. So it's very close to the end. Yeah. Okay. Okay. So this is a phytoplankton distribution, different phytoplankton distribution. This is a bigger phytoplankton, and this is a middle size, and this is a small phytoplankton. Small size phytoplankton is usually in the open ocean, across the open ocean or cross-shore area in the oligotrophic area, and the big phytoplankton you can find in the coastal area. Yeah, so this kind of change is very important for relation to nutrient, and also the eutrophication also may change this kind of phytoplankton structure, and this is also important topic of the research now. Okay, so this is the last slide. So as I said, the ocean color is maybe very useful to understand the change of phytoplankton, and which is related to the tropic condition. And but actually, we don't know so much about the ocean still. Yeah, especially we don't know how human relation, good human relation to ocean. Yeah, this is a very important question, I think. But we don't know much, and the ocean is definitely changing with influence of human. And but the question is how to manage and what is the best condition. That we really need always to have a good research to understand this, and then but we cannot wait the research condition. Yeah, so sometimes we need to have action to manage this kind of thing. Okay, that's all I have. And do we take some question time?
Yes. Thank you very much, Dr. Shizaka. So we will take some question, and I see. Okay. Second. Okay. Yeah, so if you have a question, please raise your hand, and if you rather talk, I will unmute you, and if you would like to ask question, please type your question in the chat section of the Zoom. No one is raising. I see no one is raising hand. Please don't be shy. If I don't see anyone, I might read. Okay, I have one. Okay, who is this? Okay, Li Xiaolong, you'd like to talk?
Hi, good afternoon, Sensei. Yeah, I have a question. Yeah, it's about all of us want to divide the phytoplankton into the micro, nano, and the pico phytoplankton. But if we use the pigments, we find that the absorption of the each pigments is very similar. So if we want to find a good method to derive the fraction of different size classes phytoplankton, do you have a good suggestion?
Okay, so for example, this model I am showing, this model is using actually, this is using absorption, but the whole absorption kind of spectra. It is so-called the packaging effect. So packaging effect is the absorption will change with the size of the phytoplankton. Usually small phytoplankton has more absorption, and bigger phytoplankton have lower absorption. And that can be used to separate the size of the phytoplankton. Yeah. This kind of analysis is now, it's a very, many people are working for the how to distinguish the phytoplankton group using satellite. Some people use scattering, and some people using specific phytoplankton absorption spectra. So this is many people working. And tomorrow maybe the Wong-Kuk Kim will introduce some study about the red tide detection. So this image here is not only the red tide, but is actually the including the high chlorophyll area to the low chlorophyll area. But there are some needs for the detecting red tide or harmful algal bloom, which is usually we are focusing more in the within high nutrient, high chlorophyll region, and to distinguish between the say diatom to say dinoflagellate or rhapidophyte, which is a very specific and some cases very harmful algae, and which is sometimes more difficult because the spectral, the absorption is very similar each other. All of them are, and the size is also similar. Usually they are bigger phytoplankton and belong to the brown algae. So all similar color and similar size. But still, there are some possibility that we can distinguish. Especially the next year, the NASA will launch SPACE, which is the new hyperspectral ocean color satellite. So that kind of thing, the phytoplankton group identification is a major research area for using hyperspectral maybe from next year.
Thank you. Thank you.
Thank you, Dr. Shizaka. Is there any other questions? We have 2 more minutes, so maybe accepting one last question. And if you have a question later, the material is made available again on the GitHub account, so you know the contact information of Dr. Ishizaka. You can ask him directly, but this time is really good occasion that you can get
one
answer.
Question in the Q&A.
Oh, okay.
You have mentioned that near-infrared collection affects detection of phytoplankton using.
Let me first share the question. Okay.
Can you?
Okay, so there was a question. Okay.
Can you collaborate on that? Okay. I didn't talk too much about the things, but the near-infrared near infrared. Okay. Okay. The thing is, for the atmospheric correction, we are using near infrared, and the near infrared is we mostly assume that there is no signal from water usually. I mean, or very low signal from water. But if you have a very high turbidity, yeah, even the near infrared, there are some signals in if very turbid water. Yeah. So then, if there is a signal, then there's a conflict with the algorithm, because the algorithm is assuming a very almost no signal from the near infrared region. Yeah, so that makes usually pattern chlorophyll concentration. Actually, usually it will make a problem for the spectral shape and makes usually overestimation of the chlorophyll A. Yeah, so that's one of the problems. So now, yeah, many people are working for this kind of problem. Yeah, with more sophisticated method. Yeah, the coupling between the atmospheric model with in-water model. Yeah.
Mm-hmm. Okay, thank you very much, Dr. Isizaka. If you have more questions, please contact him directly or send those questions to me. Okay. All right.
Okay.
So now we will be moving to another room. I propose to take 10 minutes break, and I see you in the different room. I will keep this room open for a bit, and I will be sending you the link to the next session. Hold on a second. Okay, I see you have the link to the next session. So please, yeah, get out of this room once you find the link. I will be waiting for you in different room.